as in further to do, I would like to start with the antibiotic resistance group. They have 6 minutes, they have maximum of 10 slides, as an Ignite talk should go.
And we're going to be presenting on antibiotic resistance. So this is the day in the life of bacteria. And obviously this picture is just a cartoon of bacteria, but notice how all the bacteria are unique and different. There are two main types of bacteria, gram-positive and gram-negative. Gram-positive bacteria have a thick peptidoglycan cell wall, while gram-negative bacteria have a thinner cell wall and an outer membrane. So bacterial growth can be broken into phases. The first, lag phase, occurs as bacteria adapt to new conditions. Eventually, exponential growth begins, followed by a stationary phase. The stationary phase is often due to a limited nutrient or factor, and the population stays relatively stagnant, as seen by the curve. Eventually, death occurs. So let's say we have a population of bacteria inside a human, and it's making the human sick. So what does the doctor do? He prescribes antibiotics. Now, in lab, we cover two main types of antibiotics, even though there are many more. First, we have Bert the Beta-Lactam. Bert kills bacteria by preventing cell wall synthesis. When the cell wall is weakened, the cell is much more permeable to water, and because of that, water rushes into the cell and it lyses. Antibiotics like Bert tend to be more effective against gram-positive bacteria because they don't have that extra membrane of protection like gram-negative. Mina, the aminoglycoside, kills bacteria by preventing protein synthesis and bacterial growth. She does this most often by interfering with translation, either by a misreading of the codons or by preventing the ribosomes from assembling properly. When translation is unable to occur, proteins cannot be formed. And a lack of proteins can first prevent the growth of current and new bacteria, and the population will eventually die. This, in theory, can affect both gram-positive and gram-negative bacteria because all bacteria have to go through translation. However, this is more traditionally used for gram-negative bacteria. And so let's assume our bacteria population is gram-positive. Bert is introduced into the system, and Bert and the bacteria battle it out for several hours, and soon enough, much of the bacteria is dead. However, some are still alive. How do they survive?
These bacteria survive because they have a mutation in their genes that has made them resistant to the powers of Bert the Beta-Lactam. This is an example of spontaneous mutation. Spontaneous mutation is when a mutation occurs independently of any action of the virus, or in this case, the antibiotic, and therefore there should be much more variation in the number of cells showing the mutant phenotype. Currently, more and more bacteria are becoming resistant to antibiotics. An example of antibiotic resistance seen in everyday life is when a person becomes sick with a bacterial infection. And John is sick, so he went to the doctors, and the doctor prescribed him antibiotics for 5 days. As you can see on the graph, John stopped taking the antibiotics on the 3rd day because he was feeling better. A few days later, John is sick again. He is sick again because of resistant bacteria inside John that no longer respond to the antibiotic prescribed. And these resistant bacteria have had the time to pass on their genes to the new generation. So now the doctor has to prescribe another antibiotic that the bacteria are susceptible to. Moral of the story, always take your full dosage of antibiotics. So we wanted to see antibiotic resistance in action, and we did in lab. There are two main ways of analyzing data for antibiotic resistance, the colony count method and the distribution method. In the colony count method, the antibiotic is incorporated into the agar itself, and you count their surviving colonies. And if there are too many colonies to count, you divide the agar into 4 equal sections, count the colonies in one section, and multiply by 4. In the distribution method, you observe the bacteria's susceptibility to an antibiotic by measuring the zone of inhibition. An important thing to remember is that there is a direct relationship between the rate of the zone of inhibition and the rate of the bacteria's susceptibility. Basically, the bigger the zone of inhibition, the greater the degree of susceptibility to the antibiotic.
Okay, so below are some important points that summarize this module. Again, there are two types of bacteria, gram-positive and gram-negative. And we also went over the 4 stages of bacterial growth. So that's lag, exponential, stationary, and death. And then finally, we applied what we learned in this unit to the real world and to medicine. Thank you very much.